Hi, good morning, everyone, and welcome to Grand Rounds this Wednesday morning. It is my absolute pleasure to welcome Dr. Paul Kluetz, who is Deputy Director and a medical oncologist, but Deputy Director of the Oncology Center of Excellence at the US FDA. In addition to assisting strategic and operational oversight of that center, he's got a broad interest in trial design and endpoint selections to expedite drug development and define clinical benefit in our cancer trials. Some of his initiatives include the creation of the OCE's Patient-Focused Drug Development Program and expansion and direction of that group's efforts to advance real-world evidence, decentralized trial designs, and digital health technology. And Paul's very passionate about all of those aspects, particularly patient-reported outcomes. He's very active in overall regulatory review of oncology products and oversees a lot of oncology drug labeling initiatives. It's been my fortune to work with him on an updating of generic labels, of outdated labels of older anti-cancer drugs across the group. He remains very clinically active and continues to care for patients and supervise medical residents at the Georgetown University Hospital. So Paul, welcome to Winship Grand Rounds. I want to thank you for taking the time and thank you for giving what I'm sure will be a fascinating and provocative talk on drug development.
Great. Thank you so much, Donald, and thank you for inviting me to be able to speak to the group today. In addition to knowing Donald and working with him over the years, I was able to reconnect with Dr. Carolyn Meltzer, who's also a former mentor of mine from medical school. So it's been really fun being able to connect. Again, my name is Paul Kluetz. I'm a medical oncologist. I do work at the FDA. I'm serving as the Deputy Director in the Oncology Center, and I'm decided to give a kind of an overview talk that's going to hit on endpoints and cancer drug development from the regulatory perspective, and I'm entitling it Crossing the Cancer Drug Development Finish Line, because when we make our regulatory decision of approval or non-approval, it really kind of marks for a new molecular entity the finish line that we're trying to achieve to get an effective drug out to patients who have cancer. And I also want to talk about generating evidence for that initial FDA approval, but I want to talk about how we can start generating evidence beyond approval, because approval is just the first step in a long life span of a drug. We can continue to learn, and we should. So again, a brief outline. I want to give sort of a very high level overview of the structure of FDA as relates to oncology product development. And then I want to dive into, for some time, endpoints and trial design, because there's been quite a bit of discussion about what the right endpoints should be in cancer drug development. And I want to give you some ideas about how we think about endpoints, not just about what are meaningful, but what are measurable and what are accurate, reliable, and least prone to bias. And then I'll talk about two areas that I think are interesting: how we incorporate patients into drug development, how we look at real world data and learning healthcare and evidence generation, and then I'll wrap it up. So broadly speaking, FDA has a very big role in regulation of products in the United States. And I put a quarter up on the slide just to show that about 20 cents to 25 cents on each dollar spent in America is on a regulated product by the FDA. So we don't just assure the safety and efficacy and the security of cancer drugs, cancer biologics, but medical devices, food, cosmetics, radiation, veterinary medicine, tobacco, so many areas. And so it's a big operation. But one thing that we do as well that I don't think is that well known is that we're pretty interested and collaborative with respect to advancing science. So we fund external research. I'm speaking about cancer specifically, but it's across all those domains that I just mentioned. And we also conduct our own science. We have the largest repository of commercial clinical trial data sets, and so we do a lot of data aggregation, exploring endpoints, exploring subpopulations, all with the intent to make drug development more efficient and understand safety and efficacy of products. One thing that we don't do that's a misunderstanding oftentimes is regulate the cost of products. We don't know how much a product will cost before we make a regulatory decision, so we don't regulate drug pricing, and we also don't regulate the practice of medicine. That is to say, when we approve a drug or a biologic in a specific indication, like metastatic prostate cancer, for instance, that can be used in other indications. It can be prescribed in other indications by a prescriber. We don't regulate how they're used. Now, companies can't market their drug for an off-label indication, but physicians, clinicians, providers can prescribe as they see fit. The centers, the key centers that regulate products for cancer are as follows. There's the Center for Drug Evaluation and Research, or CDER, and that's where a large amount of work occurs. That's the cytotoxic chemotherapies, the small molecules, the therapeutic antibodies. The Office of Oncologic Diseases, which I also hold a role in, is an incredibly busy group. That's also is also a group that has about 100 medical oncologists and deep expertise in a sort of an academic model. CBER is the Center for Biologics, and that's where cellular and gene therapies and vaccines are regulated. And CDRH, or Center for Devices and Radiologic Health, looks at therapeutic devices as well as in vitro diagnostics. And so when you look at those centers, you realize very quickly that there is a lot going on in cancer product development across all three centers. And so, interestingly, about a couple of years ago, we were set up as the first Intercenter Institute. Many of us coming from the Office of Oncologic Diseases in CDER were asked to form the Oncology Center of Excellence. This is led by Dr. Pazder, as well as a group of executive leadership that share roles within both OCE and CDER. The intent here is to create consistency in the clinical review of cancer products across those centers. And it makes sense as what therapeutic area should start this idea of a, rather than product-oriented, but more of a therapeutic area-oriented process, that it should be oncology, because oncology has so much science going on, it has so much cross-center interaction, and it's been really a rewarding opportunity to sit, at the front of this new organization and create these both, inside the FDA and outside the FDA collaborations, which has been really interesting and rewarding. I will say that the job is all about striking the balance, as you can imagine. I use this slide just to give you an idea of how I think of it. On the x-axis, it's all about certainty and data and regulatory burden, and they all go in the same direction. So, if I want really high certainty on the efficacy of a drug, I might ask for 2 or 3 randomized controlled trials with survival endpoints. You can imagine how much data and how much burden that would be for development of an individual product. So you get more and more regulatory burden. You might hear, we're being too cautious, we're stifling innovation, we want to reduce regulatory burden. On the other hand, if you move too far down to the left side and you accept a small data set for a very large population, looking to expedite development too much, you may have too small of a safety data set. Maybe you have a delayed safety finding, and you'll hear calls that FDA is asleep at the wheel. So there is a balance there on how much certainty and data we need, because that will come with burden, and where is that balance? And I feel like we've done a pretty good job at navigating that balance and continuing to learn in the post-marketing setting. In addition, on the vertical axis, how we operate has to be a balance. We have to be consistent in our regulatory decision-making, but we have to be flexible to pivot with, rapidly emerging science, as well as rapidly changing therapeutic landscapes. We have to be very thorough in our reviews. There's a lot of data to evaluate in an NDA or BLA submission, new drug application or biologic. But we also have to be very efficient, because we know that if it's an effective drug and we're going to have an approval action, time is every day is a day that a cancer patient doesn't have access to a therapy. And then we have to be independent and make our decisions based solely on scientific data presented, but we also have to be interactive outside of our individual applications. We have to get out there, we have to speak to the cancer community, patients, and organizations, do things like I'm doing now, so that not only can the community understand what regulatory science is about and what our regulatory objectives are, but we can understand from patients and from the community what's happening at the site level in clinical trials et cetera, so we can be better at what we do. So I want to spend a little bit of time, I think, on design and endpoints, because I think that's a lot about what is going on in cancer drug development right now and how it's changing a little bit. So this is the phased drug development paradigm. This is what we've done for decades across therapeutic areas. It's very sequential and rigorous. We look at non-clinical data in yellow and make sure that we have a safe starting dose, and then we bring a product, a new molecular entity, into clinical trials into patients. And so that IND dotted line is an investigational new drug application. So now you're asking FDA, is it okay to put it in human beings for the first time? We then used to spend quite a bit of time looking at pharmacology and dose as we dose escalate, looking for the right dose. We may take that into a phase 2 type of situation where we start to look at efficacy, but maybe in a couple of different doses and identify an optimal dose to then take to the blue arrow, the therapeutic confirmatory stage, which would be a large randomized trial to support a licensing application. The red dotted line here, which would, we'd make a decision based on that. Now, this can take a long time, and for a long time, this is what we did. And this is a slide by Dr. Theoret, so this has got some amazing animation. So I want to thank him for it. We've really moved to seamless oncology drug development. So what we've got now is we've got a sort of a collapse of all those phases into more of a seamless way of looking at drugs. Well, we get them into humans in the IND phase and in a dose escalation phase. We know so much more about science and about the mechanism and about the targeted agents now that we do see early signs of efficacy in a much more robust way than we had in the past, allowing us to expedite things. We also got a new approval authority in 1992 called the Accelerated Approval Pathway that would allow us to get a licensing application based on some of this earlier clinical data. And so what you have is you have efficacy, you're looking at efficacy earlier, you're moving the licensing application earlier, and now we have let's say a single arm trial with response rate that's of high enough response and duration that we feel comfortable. We're reasonably likely to predict benefit. We do an accelerated approval. There's a post-marketing trial underway, and when that post-marketing trial comes out, we then can grant regular approval if it verifies benefit. So that's all about what's called the accelerated approval pathway. So let me talk a little bit about that. So there are two approval pathways in the United States for drugs and biologics. There's regular or traditional approval, and there's accelerated approval. And which path you take depends on the endpoint, the primary endpoint of the trial, and the magnitude of the effect in the context of the disease and existing therapies. And so it has a lot to do with the level of uncertainty that we have at the time of the decision and the unmet need. So regular or traditional approval, these are typically larger trials using a time to event endpoint like progression-free survival or overall survival. They have a larger safety database. On that balancing slide, I guess you could say that we have more data, more certainty, and therefore we feel comfortable with a regular approval. The one important point about regular or traditional approval, there's no comparative effectiveness regulation. Your drug just needs to be safe and effective. If there is already an effective drug in that space, then you can put your drug up against that effective drug and show that it's non-inferior. And therefore, we have non-inferiority trials. That's sometimes misunderstood. Your drug doesn't need to be better than available therapy. The other thing to note is that there's many calls that we should only approve drugs on overall survival or health-related quality of life. That is not required for regular approval. We can use tumor-based endpoints and will continue to do that. And there's reasons for that, and I'll try to get into that a little bit later. Accelerated approval, again, 1992, based on the HIV crisis at the time, there was an authority for severe and life-threatening diseases like HIV, like cancer, to allow approval on an endpoint, an earlier endpoint that's reasonably likely to predict clinical benefit. Now, typically that's response rate in cancer trials. It could be progression-free survival if it's a small trial and there's uncertainty about the magnitude of the benefit. it has to, in this case, because there is some uncertainty, has to provide a meaningful benefit over available therapy. So here in accelerated approval, you do actually have a comparative effectiveness type of requirement. So that is why when we see a response rate come in for accelerated approval, and it's 40 or 50%, the available therapy, the FDA approved available therapy, needs to be meaningfully less than that in order for it to achieve accelerated approval. But again, because of the uncertainty, these will require post-marketing trials, like the seamless oncology slide I showed you from Dr. Theoret. And those confirmatory trials should be underway, and there's clear reasons for that. When I get an application in and I think about the results from an efficacy standpoint, I look at it in three buckets. I think about what is being measured, the endpoint itself, and how clinically meaningful is it. So survival, very meaningful to patients. There are symptom and functional outcomes that are also sort of direct measures of clinical benefit. And then there are clinically important endpoints in oncology, which are typically tumor-based endpoints. And then down the line, there are true sort of biomarker, efficacy response biomarkers like PSA, which we haven't really used much. We then look at how accurately is it being measured. So even the most, even if your endpoint's very clinically meaningful, it still may be challenging if it's susceptible to bias or it has, poor measurement characteristics, poor accuracy, et cetera. And then we look at the magnitude of the effect after we look at that. And again, this is my personal way of looking at it. This is not a structured way that FDA reviews things. This is not going to be a surprise to anyone who's on this call, given that this is a cancer center, but just to put it in perspective about when the timing of these endpoints hit, this is just a cartoon of a tumor growing, shrinking, et cetera, moving into the earliest endpoint of response rate, shrinkage of tumor by RECIST criteria. It begins to grow again if it becomes refractory to the therapy. You then get a progression endpoint like time to progression or PFS. All along this continuum, you could look at some kind of symptom or functional outcome, whether that be improvement or delay in progression of a functional decline. And then the last event, unfortunately, in the natural history of most solid tumors, is death, and we look at overall survival in that case. Now, there is a lot of controversy in the literature about, again, about health-related quality of life or survival needing to be the endpoints for cancer approval. And I think, there's a lot of things you need to think about, and I'll get to a slide on strengths and limitations of endpoints that might hit at this a little bit as well. But some things to think about our disease area that are unique compared to other therapeutic areas and an advantage for drug development. One thing is that cancer naturally progresses, and most cancer patients with metastatic tumors will die of their cancer if it's left untreated. And that's, so to know that natural history and that it inexorably grows helps us with understanding how our endpoints work. Number one, tumors don't shrink on their own. So that makes response rate, shrinkage of a tumor, very useful in a single arm trial because we can isolate the effect of the drug and we know that that outcome is due to the drug and not some other prognostic or unknown prognostic factor that we missed. Additionally, as far as the meaningfulness of the measures, tumor measurement is clinically important for decision making. We use tumor measurement in practice, and tumor growth, when you've had a patient on a therapy for years sometimes, and you see your CT scan has progressed, and you know that drug is now having resistance, it triggers therapeutic change. And anyone who's ever had to have that discussion, and we all do with our patients, it's pretty meaningful and a bad outcome. Additionally, tumor shrinkage or response rate can be very important. Could change surgical plans in the neoadjuvant setting, depending on where the tumor is located. If it's disfiguring skin lesions or if it's in vital organs, that can be very important as well. So while they're not direct measures of clinical benefit per se, tumor-based endpoints are clinically relevant, and they're certainly much stronger than something that's, say, a distant biomarker, which some other therapeutic areas have to rely on. As far as measurement characteristics and how an endpoint's measured with respect to variability and bias, I think this is important. It's an important aspect for people to think about because, again, just because you have a very meaningful outcome, if you can't rely on its measurement characteristics and you may have a false positive or even a false negative, that's not going to help drug development or patients. So as I think about interpretation, I just use a couple of common endpoints. So the lowest interpretation required obviously would be overall survival. The event is death, it's binary, it's easy to interpret, you know it to the day. So very low risk for bias at the endpoint itself. Measuring tumor progression when it's measurable, like a RECIST type of progression, is objective. There is a little bit of interpretation. We all know that there are some differences between investigator and blinded review, especially right around the gray zone of the progression event. But it's even more challenging for tumor progression in bone, the third from the bottom bullet point. I do a lot of prostate cancer work, and so I was working a little bit with the Prostate Cancer Working Group 2 and 3, who created a way to look at progression in the bone for prostate cancer, which has been a useful progression endpoint, but it's complicated, and it does require interpretation of bone scans, which, as everyone has seen, a bone scan can be challenging. And then at the highest level, you start getting into events that are driven by decisions that are made at the clinical level. And skeletal related events might be an example of giving radiation for a painful bone met. I mean, you have to think about what happens before that event gets triggered. The patient has to tell the doctor they're in pain. The doctor has to believe that it's focal enough that it could be cancer, maybe do a scan or something to identify and believe it's cancer, and then make the decision to give radiation and when to give radiation. So all of those things give you a lot more risk for bias. And this extends to almost all of our delay and morbidity sorts of endpoints. You can think of bladder cancer and delaying or making a cystectomy something that doesn't need to happen. That's a really important benefit to patients if you can delay cystectomy or avoid it. But that decision whether to do a cystectomy has to have some guidelines on when and how, or when to do it and whether to do it, because if it's just up to the subjective decision of a surgeon, that could introduce a lot of bias. I mean, the summary of this is that there's no free lunch. So there are strengths and limitations of every endpoint that we use, and ideally we'll be looking at more than one, and we do. But you can see if it wasn't for feasibility, and I'll talk a little bit about that, I think overall survival would be the gold standard endpoint because it's clinically meaningful and because it has a low risk of bias. But increasingly, it's becoming very hard to achieve a clear overall survival endpoint result given crossover and given some of the equipoise challenges we have, which I'll talk about. Tumor endpoints, as I mentioned, are sort of plus minus on clinically meaningful. They're clearly very feasible and commonly used, and the risk for bias, I think, is relatively low. Symptom endpoints, very clinically meaningful, quite feasible, but plus or minus on risk for bias. It's a subjective endpoint, therefore has a risk of placebo effect, et cetera. And I believe that those endpoints are important, but typically are more complementary to understanding what's happening with the tumor. And the way that I talk about this, because it's actually an area of interest of mine, is that if we got an application that was solely based on an improvement in pain, and there was no evidence of anti-tumor activity, it really might not be considered an anti-cancer agent, more of a supportive care agent. And if you think about what your level of acceptance for safety would be for a supportive care med like morphine compared to any kind of anti-cancer agent, we would have a much different risk-benefit tolerance for adverse events. And so I think symptoms and functional improvement are typically used to support a tumor-based endpoint in cancer therapies. Finally, that last row is what we were just talking about, clinical outcomes, reduction in healthcare utilization or reducing morbid procedures. Very meaningful, feasible to measure, but pretty subject to bias unless you have really strong guidelines on the decision making. So what's being measured is important, how accurately it's measured is important, and then we look at the magnitude, and the magnitude cuts both ways. If you have a large magnitude of benefit, it can certainly overcome uncertainty about meaningfulness. So for instance, progression-free survival, if you have a 2-year progression-free survival benefit, you can feel pretty comfortable that that's significant. Whereas a smaller benefit increases your uncertainty. And then, on the other side, small magnitudes can be clinically meaningless, even in the strongest endpoint like survival. And so, we take that into consideration. Last point I'll make about interpreting a result is that just because you get a huge benefit in survival or a huge benefit in progression-free survival in a randomized trial, it doesn't mean that it was due to the drug. So, the next step we all do is we really turn on, in that journal article, to the prognostic factors and balancing and demographics and, how severe the diseases were at baseline for each arm, because we know that imbalances in prognostic factors can lead to, a false conclusion that this was due to the drug. In addition, I think maybe even more challenging for us as we get many more available therapies is subsequent effective therapies, and any asymmetry in that can lead to a false conclusion. So I would just end by saying randomization is so critical to balance known and unknown prognostic factors for time to event endpoints like survival or like progression-free survival. And we just don't feel like these time to event endpoints are interpretable in a single arm setting. So we do rely on response rate. So I will start my next segment by just honing in on response rate, why it's such an important endpoint in oncology, especially now. Again, tumors don't shrink on their own, so we have a high certainty that tumor reduction is due to the treatment. Now, caveat, if it's a monotherapy, a single agent that we're investigating, it's even better because we know it's the drug we're reviewing caused the reduction in tumor burden. But if it's a combination therapy, and that's becoming increasingly happening, the contribution of the new drug versus what you're adding it to is a challenge. So, you do have to watch out about combination therapies with these single arm trials. So we can use them in single arm trials, and that's increasingly important for reasons that I want to get into regarding really precision medicine. The other huge benefit of response, as everyone knows, is it's the earliest event in the natural history of the disease, and therefore these are outputs that you can get quickly and also with less patients. So, where have we been and where are we going with endpoints in oncology? So in the 70s and before, this was a setting of very limited available therapies, and response rate or tumor shrinkage was accepted as a primary efficacy endpoint for regular approval. At that time, we didn't even have accelerated approval, and drug development was generally using that. And there were randomized trials, but in general, there was a lot of response rate. In the 80s, reading back through advisory committee meetings, this is when we really started to see some combinations being added, increasing, dose of cytotoxics and getting a lot of toxicity and maybe getting more 10 or 20% more response rate out of it. And it was really thought that given the toxicity, there's really a lack of clarity as to whether that level of response would translate into longer term benefit for patients. And at that time, it was felt that measurement should really reflect survival, direct clinical benefit. And we went into an era of a couple decades of large randomized trials with overall survival endpoints. And I think this idea of does 10 to 20% response rates, will that reflect long-term benefit, has come up recently again, and we'll talk about that in a moment. And then in 2000, we started to see the advent of, I guess we'd call precision medicine, and this great paper by Dr. Drucker showing this incredible 53 of 54 patients with complete hematologic response. And in this paper, they end it by saying our results demonstrate the potential for the development of anti-cancer drugs based on the specific molecular abnormality present in human cancer. And boy, when you read that now, you think, holy cow, that was really an understatement, and how this has actually just been an incredible targeted therapies, and really understanding the biology and the basic science has led to such amazing results for our cancer patients. And it is a double-edged sword. So there is something called clinical equipoise, and equipoise means that there's a general uncertainty with the expert medical community whether a treatment is effective or not. So if you have equipoise, you really don't know. And so when you look on the left to the table that's the oxaliplatin drug development table, when you have a 9% differential response rate, you feel comfortable that a randomized trial would be appropriate, and that allows you to feel that the trial is ethical and feasible from an enrollment standpoint. On the other hand, when you start to see 50 to 60% response rates, like you see in crizotinib on the right in non-small cell lung cancer, and the available therapy has a 20% response rate and it's a doublet chemotherapy, you start to think, do we have Equipoise for this randomized trial, especially if the endpoint's survival and you don't allow crossover? You know, that really becomes a challenge, and that's been the challenge we've been facing. It's a good problem to have this kind of activity, but it does create challenges. And the challenge continues. This is a slide by my former colleague at FDA, Guidan Blumenthal, showing just the vast array of targeted therapies in the lung cancer, non-small cell lung cancer space, with big differentials in the available therapy at the time, chemo versus the single agent targeted therapies. Not only is it about big effects and loss of equipoise, but it's about small subsets. So 20 years ago, your non-small cell lung cancer pool of patients for our large randomized trial was everybody, essentially. And today, although this is an older slide, so there's probably more cuts in the pie, and I know there are, you know, now you've got a lot of small subsets, and it is harder to accrue 1 or 2 or 3 randomized trials if you have competing drugs in that area in 1% of lung cancer. And so that's been a challenge. As we wrap up endpoints, there's been a lot of discussion as to whether progression-free survival or response rates, you know, really are helping patients, you know, at the population level. And, you know, we've had so much activity in non-small cell lung cancer and drug approvals, many of them based on tumor-based endpoints. I found this paper interesting that Dr. Howlater and colleagues in the New England Journal looked at 2-year lung cancer survival in non-small cell lung cancer over that time period of this you know, huge amount of targeted agent approvals, and saw about a 9% improvement in men and women in mortality. And you know, you might say they did try to adjust this for all sorts of factors, but you may even say after all that adjustment, well, maybe it was just better screening, maybe it's better care, maybe there's all sorts of reasons why this may have occurred. How do you know it's the available therapies? Well, they did look at small cell lung cancer and notice, you know, not the same benefit in this therapeutic area that has had very little, unfortunately, very little effective drug development in that time frame. And so it was sort of an interesting control. So, you know, response rates are fine and single arm trials are good, and they've certainly benefited drug development and accelerated approval and targeted therapies. But despite the importance of response rate, accelerated approval of immune checkpoint inhibitors particularly has been a bit of a cautionary tale, and that was a tale misspelled there, unfortunately, but that's a tale that's been told at the Oncology Drug Advisory Committee recently, where multiple checkpoint inhibitors granted accelerated approval with response rates less than 20% did not have successful post-marketing trials. It could have been for a variety of reasons, but the fact remains that they didn't achieve that, some of which have been voluntarily removed from the market. And so what that really showed us is that, you know, again, it's always a balance. And when you have these modest response rates, particularly less than 20%, if you're considering accelerated approval, you know, the randomized trial, you need a randomized trial to show that that's really going to benefit long term. And it should be well underway and preferably fully accrued at the time of accelerated approval. It'll be a stronger accelerated approval application if that is well thought out. Some thoughts include starting that randomized trial and fully accruing it, and then maybe submitting the accelerated approval based on an early look at the response rates within that trial. So that's endpoints and trial design. I wanted to get a little bit into some of the work that we've been doing in a couple of areas that have been of interest to the community. So one is incorporating patients into drug development. And this is a slide that I got from Teresa Mullen from the FDA when she was describing what patient-focused drug development was all about and what FDA was trying to do to achieve that goal. And really what it is incorporating patients all along the drug development continuum to essentially create evidence packages that mean something to patients, and to be able to communicate information to patients and their prescribers better on how to use these drugs. Now, there's all sorts of areas within drug development where you can insert patient feedback. One of them is translational. Before you even start development in a particular disease space, you could ask patients what matters to them most and can it be measured, as I was mentioning about the challenges with measurement. In the clinical studies space, we can then seek to incorporate some of these endpoints that matter to patients, and also just look at the trials themselves and try to make them more patient-friendly and least burdensome. In the pre-market review space, we are looking about how can we analyze sort of symptom and functional data, for instance, and incorporate that better in our reviews. And in the post-market setting, we need to communicate that information, that patient-generated data, out in a way that can help patients and providers make decisions. And we got to keep getting more information in the post-marketing setting. And it's more and more important as our preliminary trials are usually one trial and sometimes single-arm trials. We need to generate more data in the post-marketing setting. How can we do that efficiently and effectively? So up front, I think we've done a lot of work in this translation in clinical studies area. I would say the first thing when we asked what matters most to patients in cancer control and other than cancer control and survival, because I think when you ask patients, mostly they'll say, you know, I'd like to be cured. And if I can't be cured, I'd like my cancer to be under control and increase how long I live. But regardless of all that, they would really like to know how they're going to feel and function when taking their therapy. And a lot of this has to do with tolerability more than, you know, improvements in symptoms and function. They're worried about the side effects of cancer treatments. And I think we could do a better job with that. So there's a resurgence of interest in patient-reported outcomes and measurement of symptoms and function using other ways like wearable devices. Why now? Why do I think it's resurging? I think it's because of several important factors. One is I think we have so many more available therapies, and that's a great thing for patients. But both sponsors and patients now want more ability to discriminate between, say, two different therapies in the same line. And I think having more informed choice means having more data on safety and tolerability. And I think we're going to see a little bit more comparative tolerability types of designs moving forward. Technology is improving capabilities without a doubt. Electronically capturing patient-reported outcomes, for instance, is more common. And now we are starting to see wearable device data and how we can look at that. And we have a couple of prospective research projects underway in that space. I think clinical care is starting to use patient-reported outcomes more. I think that's going to really start to make it more common in trials as well. I think Ethan Bash's work showing that if you can look at symptoms through patient report and monitor them and act on them, you can improve outcomes. I don't know how much of that's going to, you know, become commonplace, and if it does, then of course it will be commonplace in trials as well, because we take care of patients in trials. And then finally, item libraries means that the tools that we use, the patient-reported outcome surveys, have gotten a lot better and a lot more flexible, so that when you have, you know, very different mechanistic drugs that you're comparing, you know, you can really select the expected toxicities rather than use a kind of one-size-fits-all form that may be missing very important, relevant symptom or functional deficits. So what we did a couple of years ago was just initiate a sustained effort to try to make this information more useful for regulatory purposes. It had to meet a couple of key criteria. It had to be important to patients, what we're going to measure, but it also had to be sensitive to the intervention. That means sensitive to the toxicities of the product that we're looking at, but also sensitive to changes in the status of the patient, whether with respect to disease. And so we come up with 5 core patient-reported outcome concepts that we would like to see more commonly, and that would be disease symptoms, a selection of the most common symptomatic side effects, a single question about the global overall impression of the side effect burden for the patients. One of them is the FACT GP5 question. I'm bothered by the side effects of my treatment. That could give you a nice overall read. Physical function, both by patient-reported outcomes as well as wearable devices, is of interest, and then role function. You know, can patients still work, or if they're retired, perform their leisure activities? And I think while this isn't everything you could measure about health-related quality of life, of course it's not. This is a good sort of minimal expectation and really covers, I think, aspects of the patient's quality of life that is most likely to be affected and move with a drug intervention or a disease. So what I'd like to see in the end is just expanding our totality of evidence that we get to review for safety and efficacy of a drug. Not only will we have what we do well right now, which is in the blue and green, survival, tumor measures, biomarkers, clinical safety data, but also in the yellow, could we do a better job at organizing how patients are hospitalized, or how many ED visits they have, or morbid procedures they've had to undergo, or supportive care use. This is pretty easy to capture, but it's not done in a systematic, standard way. Could be done better. And then the patient-generated data also could complement this. These are just the core outcomes that we mentioned previously. To hit this home, we did just release a guidance to industry. Guidance to industries are important documents that sort of can give FDA's current thinking on a topic, and this, a lot of what we've talked about with patient-reported outcomes is in this guidance, and I refer you to it with this link. What about trials themselves? How can we make them more patient-friendly? There's been a lot of work in this, and it's a very exciting area. Number one, broadening eligibility criteria there's a lot of support for that, and we've worked with ASCO and Friends of Cancer Research on that, looking at what have we been doing with eligibility criteria? Does it make sense? It's just basically putting a lot more thought into why are we excluding patients, and can we include more patients in trials? I think improving trial access through eligibility criteria or decentralized trials is going to have an opportunity for us to reduce disparities. It could actually improve the accrual of subgroups that we've had a hard time accruing before. And so we'll talk about decentralized trials. Those are driven by improvements in digital health technology, tools that can remotely assess patients. We'll talk about that a little bit. And all of this together, digital health, decentralized trials, is pushing us towards doing a better job with learning from our healthcare systems. And if we can get our healthcare system data in somewhat of an organized situation, we may be able to deploy pragmatic, practical trials in a much more easy way than we can now. So, but let's talk briefly about decentralized trials. So decentralized trials just mean you're moving some or all of the conduct of a clinical trial outside of brick and mortar sites like Memorial Sloan Kettering or MD Anderson and out to where patients live in more of their natural healthcare environments. You can do it all, or in some cases in a hybrid way. What are the benefits of this? Well, clearly you'd reduce burden on patients for travel. If you could improve accrual because you have wider access to patients, it could lower costs and make trials faster. If patients feel less burdened on a trial, they may stay on a trial, so decreased attrition. We can assess patients in between clinic visits because they're not going to clinic in some cases, and therefore maybe you have more frequent assessments, and the ability to do assessments easier may allow you to follow up in a longer term manner, which has always been a challenge too with cancer trials. And all this could definitely make our populations more representative. But there are challenges and barriers to decentralized trials. There's uncertainty and risk aversion, probably first and foremost, because it's an expensive clinical trial environment that we're in. And uncertainty and risk aversion is part of that whenever you spend a lot of money on something. Then there's things that FDA has no control over, like jurisdictional laws and telemedicine licensing. I think training of local healthcare providers to the extent they need to be trained is important, and how you supervise remote DCT personnel. But I think the most important challenge and barrier is this first bullet. How much uncertainty are we instilling in the data? And does remote trial conduct add substantial variability to data, especially data on safety or on the primary efficacy endpoint, which is often a radiological endpoint? So we have, of course, COVID-19, unfortunately still not through that yet. And what has really happened is we have moved to a hybrid decentralized clinical trial in most interventional cancer research, especially commercial trials. And so what we need to do now is learn about the effect, if any, on data quality in some of these remote assessments. Telemedicine, remote labs, remote imaging, getting the product out to patients, remote administration of investigational product, and remote site monitoring. And in order to do this well, we recognize quickly that we would like to see data sets when they're submitted to FDA to be flagged in a standard way on which assessments were remote and which assessments were standard clinical trial assessments. So we've stood up a website looking to advance this. We've spoken to regulated industry about this, and we're hoping that we'll start to see data sets come in a way that's flagged so that we can help generate the science and de-risk these sorts of designs because we think they could be useful. Lastly, is the question of generating more data in the post-marketing setting. So real-world data is a huge buzzword. What is it? Real-world data is simply data collected about a patient's health status or the delivery of healthcare routinely collected from a variety of sources. It's essentially data collected outside of a standard clinical trial, and you can get it from claims data, EHRs, registries, patient-generated data through electronic PRO, et cetera. But what's critically important is that real-world data is not real-world evidence. Real-world evidence is evidence regarding the use or potential risks and benefits of a medical product that's derived from the analysis of real-world data. So right now, we probably really need to be talking about real-world data because there has not been a lot of evidence generated yet using this data because it's challenging and because we're still working through it. So how might we use real-world evidence to help us understand regulatory issues? Obviously, we could look at use data. How often are patients using a certain drug and when and in what populations? That's something that we do now. We also look at real-world data for safety. We can learn more about the safety of a cancer product. Natural history starts to bring in outcomes, and that starts to be where we have some challenges. You know, how do you understand the natural history of a patient population? You know, how long do they live? What sorts of therapies do they get? And then, you know, where everyone wants to go right away is efficacy. Can you generate information in the real world that makes you confident that a drug is effective in a different population than it's indicated? And we're doing a lot of work to try to answer that question. In the end, I think we are seeing a blurring of the lines between randomized controlled standard clinical trials all the way over to retrospective real world data. And you can walk down the line and things change. I think right now, you know, we are a little bit more in the hybrid decentralized area right now where we have prospective randomized trials that are using some standard assessments and some remote assessments. These are highly monitored trials. Maybe the population can be a little bit more broad because you could move these out more to where patients live, but they're still generally, you know, a sort of adequate and well-controlled trial. As you move over, maybe we could do less monitoring and maybe we could do fewer assessments or just have the assessments be how normal healthcare is delivered. And that would be something like a pragmatic trial. And then finally, all the way over to real world data, these are usually retrospective currently, could be prospective, often not randomized, what we have been receiving. No real standard assessments, it's just clinical care, not monitored, and maybe the most broad population. But you can see it, depending on your research question, you might use any one of these, but we are right now in at least commercial cancer trial development in this area, RCTs and decentralized trials. So in conclusion, and I want to save some time for Q&A, the goal of drug development from the FDA perspective is to provide substantial evidence that the drug is effective and that the magnitude of the effectiveness that we see is acceptable in the setting of the drug's safety profile. When we look at an efficacy endpoint, it is not so simple as just, you know, let's measure survival or quality of life. There are many aspects of an endpoint that are important, and that includes feasibility and measurement characteristics. We do have to take care in a randomized trial that we assure that the outcome is due to the intervention and not some known or unknown prognostic factor. And so randomization is so important for time to event endpoints. We can and have been making trials more patient-friendly to complement our existing clinical trial results with symptom and functional measures, as well as broadening eligibility criteria. I think in the end, we're going to continue to explore ways to make evidence generation more efficient because we need more evidence, especially in the post-marketing setting. And so we have a lot of work ongoing right now to look at improving real-world data, at improving trial efficiencies, improving registries, et cetera. And I look forward to collaborating with the community more on that. So I do just want to have a little brief acknowledgement slide. That's some of the many people that I work with a fun picture. You know, the Oncology Center, the Office of Oncologic Diseases, very hardworking divisions at the FDA that review these cancer drugs very smart people and a joy to work with, particularly some of the leadership, Rick Pazder and Mark Thierry, Julia Beaver, Tammy Kim, have been, incredible to learn from and work with. And then Vishal Bhatnagar runs the patient-focused drug development program. Donna Rivera runs our real-world evidence program, and they've been fantastic. So thank you for your attention, and I'm happy to take some questions with Donald.
Thanks, Paul. Fantastic talk as always. Really appreciate your perspective from both FDA and your clinician hat as well in terms of drug development and how we might see some changes over the future, and particularly in the patient-reported space and other areas. We've got a few questions that I'll try to make sure that I interpret correctly, and coming from the audience. One is, when investigators apply for the IND, who would be better to file that IND? And so here's I think what the question's getting from Dr. Shin. That's if we are working with investigator-initiated trials at Emory, and we are in discussions with a company to say, all right, we've got an IND for a compound, and the company is suggesting we do it, or we would prefer the company do it. Is there an FDA preference on that? Or is it simply, look, there's an IND and we just need to make sure that the holder does what the holder needs to do as the sponsor of the trial?
That's an in-the-weeds question, Donald. But I would say that in general, we need to have an investigational new drug, an IND filed, and I don't think we, especially if it's a small investigator-initiated trial, that we feel strongly one way or the other. I do want to mention that not all investigator trials especially of approved products need to have an IND. So make sure you are aware that there is such a thing as an IND exemption, which saves some time. Yeah.
Absolutely. That's almost my number one goal for writing a new trial, is to try to get an IND exemption. Another question from Dr. Keller. Has the content of informed consents in your mind changed over time to improve enrollment and patient knowledge? And perhaps this gets a little bit to learning as we go, and more integration of real-time information in the next phase of drug development.
Yeah, I think this gets outside of FDA a little bit with IRBs as well, but all I can say is that I have reviewed consents for family members. I don't know if we're going in the right direction. I mean, I think they're very long. They're very challenging to read. And so I don't know what the right answer is for that. I think that we have a lot of room for improvement in being able to converse with our patients and adequately describe the risks, but not throw every possible thing at them. I think that really worries patients, at least the family members and people that I've worked with.
Yeah, no, that's fair. Here's a great question from Dr. Lawson, and a provocative one for you potentially. Thanks for an excellent talk. Are there data on patient-reported outcomes in individuals receiving placebos?
Oh yeah, for sure. Yeah, I mean, we have, although it's mostly add-on trials that we have placebos now. I mean, it's kind of rare to have a straight-up placebo randomized trial. But yeah, to your point, one of the great parts about working at the FDA and why we've been able to advance a lot of novel ideas with respect to analyzing patient-reported outcomes, for instance, or investigating open-label bias, which we've done some, is that we do have these trials, and they have done patient-reported outcomes with them. Now, many of the trials in the older era, 5-10 years ago, they were just throwing them in there, and I don't think they knew what they were looking to ask. There wasn't really good, clear research objectives, and the data quality wasn't so great, so it was hard to answer that question. When I think about placebo effect for PRO in cancer trials, my thought is, and we have not yet been able to see a sustained sort of false hope bias, you could say, that's sustained. Because if you think about it, if you have a drug that really isn't working in metastatic cancer and you're giving an agent that's definitely got side effects, any improvement in quality of life that they have is because they're excited to be on an investigational drug, it's probably going to be fleeting, just looking at it from face validity. So we continue to look at that. We have not yet seen a strong, consistent placebo effect, but there's a lot more work to be done. We know placebo effects happen in pain trials, obviously.
Yeah, no, I think yeah, that's a good point. You would expect an erosion of that effect to be pretty fast and pretty complete within a given patient's experience on a trial. Another question from Dr. Keller around a non-oncology topic. Based on everything you said about oncology drugs, would you have any willingness to comment on the FDA approval of the new drug for Alzheimer's?
Yeah, I don't think I'm going to be able to give a talk in the next 5 years that's not going to have that question thrown at me. And thankfully, I don't work in that space, and I just don't have that much information about it. And I have, as everyone does, I have my own personal opinions, but I don't think it's the appropriate place to express them.
Yeah, I think that's fair and understandable from your perspective. Paul, I had a question actually as well, thinking about inclusion and integration of more tolerability data and adverse event data. Two questions at once. One is, when you look at some of the real world trials that have come forward, let's take the TAPER trial from ASCO, they're only interested in reporting grade 3 and 4 adverse events.
Yep.
What is your personal, or your take on that globally? And when I look at grade 1 and 2 adverse events, I find many of them to be clinically meaningful for patients, particularly, to use a pharmacokinetic term, as the area under the curve goes longer. If you have a grade 1 adverse event that lasts for 9 months and it's nausea, that's probably worse than a grade 4 thrombocytopenia in cycle 1. So what's your take on grade 3, 4 only trials, or how you think about grade 1 and 2 in the overarching contribution to a safety profile?
Yeah, I mean, I have a pretty strong opinion about this. I think that the idea, it makes sense for clinician reported safety data. I don't, because I think if you know a lot about a drug, it's been out for 5 or 10 years, and you're not going to find out much new on the high grade events. I do think that the grade 1, 2s that are have been clinician reported, the symptomatic adverse events essentially that you're describing, are critically important, and I think we have a really good opportunity to move that into the electronic PRO, and that's happening to some extent. But I think that's where it should be to begin with. I mean, I think that we already know that we underreport severity and incidence of symptomatic adverse events, and we also have shown, this is germane to your neck of the woods, that even with exposure response, when you're looking at PK data and you're looking at optimizing dose, we seem to be seeing better sensitivity when you look at the PRO symptom data, let's say it's diarrhea, than the clinician, because it's really capturing a lot of frequency and signal on the low grade diarrhea. By the way, grade 2 diarrhea, look it up on CTCAE. I don't know if I'd call that low grade if I was.
That's an incredibly important point. And when you think about all of this, should we be taking the investigator out of the equation at the level of the clinical site for a lot of these subjective adverse events? Because at some point, the individual experiencing the event, I mean, that's the true knowledge. That's the gold standard. And if we interpret it incorrectly, which has been shown time and time again, Ethan's work in lung cancer shows that patient data versus our interpretation of that data is different, and certainly not measured in the same frequency. Is there a role to just have an electronic capture in the patient's hands that goes directly into the CRF, and the clinician deals with the adverse event management and symptom management for intervention, but that the purest form of the data itself comes directly from patient to CRF?
I mean, I've always advocated that would be, I don't know about exclusively. I think there's a lot of discomfort because I don't think the systems are so robust and in place yet to completely decouple that. I think for the time being, while we get the ePro up and running and we feel good with the technology, I think it's good to have the clinicians also reporting it. But if we end up coming to a place where we have it really basically duplicating it, it may be that the science shows us that we can help clinicians out a little bit by not having them, do all that reporting and let the patients describe it.
Yeah. All right. One final question again. Thanks for great talk from Dr. Nuka. Thanks for your time. One question about endpoints. The progress in some cancer is phenomenal, and Nuka's in myeloma, so you and Bhatnagar can banter this way. Right. What the assumptions today, null, alternate, et cetera, to design may not be appropriate when the trial is mature. and ready for regulatory review. Are there any adaptable designs to allow for newer data to plug in dynamically as data comes in that might address this issue in the future? And I think the question is specifically around the endpoint of, an alternate designs for improved endpoints for phenomenally active drugs. I presume he might be getting into the world of MRD and some of the other very early markers of drug activity that could then be surrogates for a faster readout.
There's certainly a lot of work being done in MRD, especially in multiple myeloma, but in other areas, there's interest in circulating tumor DNA. At Julia Beaver, my colleague, is very, very active in that space. I think it's early right now. I think one of the points that he makes is, can we adapt trials as the therapeutic landscape changes? That's challenging to do. We really don't like it when trials are adapted. But what we do have to understand, because this has been a criticism, is that, why are we using this inferior, obviously not standard of care control arm in some spaces? It may be that was the standard of care. It's so rapid drug development right now. So you really have to understand when a trial starts to realize. And then we typically will get that data and we'll make a decision based in the knowledge of what's happening then. And, they did the best they could with what they had at the time.
Yeah, no, I think actually myeloma offers a good example of that. I mean, doublet comparisons these days for induction really, don't make much sense because that's not standard of care in the myeloma space these days, as opposed to triple drug regimens for induction. One last question, if you've got the time. It's 8:31. We're a little bit over. Follow up on the placebo question. We not uncommonly have patients who report long-term effects that they attribute to the drug, but we don't have necessarily data to support or deny that. Chemobrain comes to mind as an example for that. Is there a way to perhaps discern that or have a better understanding of that from a PRO perspective?
Oh man, I think attribution, which is a little bit about what this gets at, is really hard. I think attribution of drug versus disease versus something else has always been a challenge. I will say, I think at least for short and medium term safety assessments or tolerability assessment, PRO offers the advantage of a baseline. So every patient gets their baseline, and if they have, pretty significant diarrhea at baseline, and they have a little bit less significant diarrhea, if in the way we've been doing it is we'd actually call that grade 2 diarrhea because we don't have a baseline in our clinical CTCA world. With PRO, the way we have our Project Patient Voice website, we correct for baseline, so that wouldn't even be captured. If you're improving from baseline, it's not an adverse drug reaction. So attribution might be helped with PRO because it has a baseline. Longer term toxicity like chemobrain, that's going to be hard because I don't think they probably will say they have that at baseline, but it's interesting. We could look at it.
Yeah. I won't get into the attribution world of 1 to 5 versus 0 or 1 in data. I think that's probably too far into that space right now. Well, again, Paul, thanks a lot for this talk. Really great one as always, and appreciate you taking the time for all of us here at Emory and any others who might have joined in the Zoom sphere. Take care, my friend.
Always a pleasure. See you, Dr. Harvey.
Take care.